Hello, everyone. My name is Meenu, and I'm a bioinformatics scientist at Dr. Lawrence Lab at the University of California, San Diego. And today, in collaboration with David, LK, Matt, and Alex, we have designed this workshop on biomarker discovery using integrative analysis.
Are
Sorry,
you sharing slides?
I'm just about to share.
Oh,
Yes.
okay. All right.
Can you all see my screen?
Yeah, looks good.
Okay. So yeah, so the topic of this workshop will be biomarker discovery from extracellular RNA-seq and tissue of origin from GTEx RNA-seq profiles. Today we will talk about the Common Fund data ecosystem, the genotype tissue expression or the GTEx project, extracellular RNA communication consortium, and we will also share a use case using these two data coordination centers. This workshop is being recorded and will be available to all the registrants at the provided Google Drive link. So here is a link to that as well. And you should have received this via the email communicator as well. Just a few housekeeping things. We will, as mentioned, we will be seeing how to download the data from GTEx, as well as the xRNA Atlas, and conduct an integrative analysis of the two types of data. We will be covering quite a lot of material, and also the number of participants will be quite a big number. And so we do request that you post your questions in the chat box, and we will do our best to answer them. Also, make sure that your mics are muted unless you have a specific question. We will also have a 10-minute break or a 5-minute break after the downloading and analyzing the GTEx data portion of our analysis. And then we'll continue from that point on. Now, however, what we will not be able to help with is how to install the Jupyter Notebook or R or any of the packages that's being used. Okay, so any questions already, or if not, I can start with the presentation. Sorry. Okay, so what is the NIH Common Fund Data Ecosystem, or the CFDE? It is intended to provide an infrastructure that enables the broad use of the Common Fund data, and that contains a wide range of diverse data sets. When it is fully functional, it will include a portal that will be the access point to several tools that can be used to compute on the data in a cloud environment, making the Common Fund datasets fair, which is findable, accessible, interoperable, and reusable. Here is a graphic showing the different data coordination centers that feed into the CFDE. There are several types of data present here, and some of the examples are the GTEx, which contains the tissue-derived long RNA-seq data, the xRNA Atlas that contains biofluid-derived extracellular small RNA data, seq data. And you also have the HubMap, which contains the single-cell RNA-seq data. And for example, another one is the metabolomics workbench, which contains the metabolomics data. This is just a few of them, and as you can see, you've got several of these around. So how do we access these data? Each data coordination center is different. And so in this particular workshop, we will learn how to access the data from two of the data coordination centers, namely the GTEx, which is the long RNA-seq data for the adult tissue samples, and the small RNA data from xRNA Atlas. And we'll also do integrative analysis of these two data sets. The link in your Jupyter Notebook will take you, if you have not yet opened your Jupyter Notebook, you can try opening it right now, but I just wanted to show you here there's a link over here that you can click on, and that takes you to the CFDE workbench. And there's a wealth of information here that you can explore later on if you're interested. What I specifically wanted to point out is that when you come to this page down, and scroll down, you've got a place where you can see all the data coordination centers that feed into this ecosystem. And you can click on each of those to then take you to the portal that is for that data coordination center. So we'll look at all this a little bit in detail just in a few minutes. So back to our notebook. We will then look at a use case. In our case, we are going to be seeing if there are miRNA or microRNA that are differentiate between male and female samples, and if there are any correlations between these two types of data. So for this, we'll get the bulk tissue RNA-seq from GTEx. In this case, again, we will only concentrate on the heart samples. And then we will do a differential expression to look at the genes that are differentially expressed between male and female samples. We will also then get the extracellular RNA, small RNA-seq data from, again, male and female samples, specifically the plasma samples, and then identify microRNAs that are different between the two. So our analysis is going to be done in 3 parts. The first part over here is the GTEx data. So as I said, we'll download the data, we'll filter it to contain only the heart samples, and then we will normalize the data and also do analysis using DE-seq2 to look for differentially expressed genes. And then we will look at a few methods to visualize these genes that are differentially expressed. The second part is the microRNA, mRNA target prediction. So for this, there is a package called the MIR-TAR RNA-seq that gives all the possible combinations between the miRNA and the mRNA interactions. This is not an experimentally verified data set, but it is the predicted data sets. And so we will use this today. We will not use the exact package, but there is a data frame that has all these interactions already listed. So we will download that interaction matrix and then filter for the specific interactions that we are interested in based on the differentially expressed genes that we identified in the first step. And then finally, we will be downloading the miRNA expression matrix from the xRNA atlas as well as the metadata. And again, filter the data set to contain only the healthy plasma controls. And then normalize that expression, and then finally visualize the microRNAs that were associated with the differentially expressed genes from the first step. So if you haven't already opened the Jupyter notebooks, this would be a good time to do so that you can follow along. I can give you a couple of minutes for you to get that running. In case you are not using Jupyter Notebook, there is also an HTML file. Let me just show up over here. So if you go to the Google link over here, there is also an HTML file that says 2024-0909 CFDE use case for HTML.html. So if you click on that, you should be able to follow along. So all this information is already in that HTML file. And also there is also R script. So this one, the last one that shows today's date with it says for workshop.r. So there's an R script for this. Again, for those who are not using Jupyter Notebook and maybe using either the terminal or R Studio, you can use the script that's present over there to run the analysis portion of the workshop. Okay, so I hope you have opened your Jupyter Notebook. So let's move on. The first part is the GTEx data download. So there's a hot link over here to the GTEx portal itself. So it takes you to the same page that I showed you earlier from the CFDE page. So let's click on that. And what you can see over here is on the top left corner over here is a downloads button that you can access the open access data or the protected access data. Today, we'll just be looking at the open access data. And then within that, you're already in the adult GTEx, so that's adult human samples. And we will download data from the bulk tissue expression page over here on the left. And we will download the very first file over here. Just to let you know, all these files have already been downloaded and presented to you in the Google Drive. So if you have already downloaded all the files from that drive. You do not need to do this again, but you can if you have not done this before. Okay, so you can download that. And you also need to download the metadata file. So this is over here on the right side. Click on that, and here you will download all the 4 files in this section using the download buttons over here. So once you're done with that, and then here are just screenshots of how that looks if you need to follow through this later on. So then you download the expression data, as I mentioned, this particular file, and then the metadata, all these 4 files. So again, all of this is present in your Google Drive. Please do try and download all this to your personal computer and keep the folder structure the same, because the code does depend on the folder structure for it to work. Do we have any questions so far before I actually start the data processing?
If any questions, you could unmute yourself and ask.
Okay, if you had any questions, please do put it on the chat as well. We can continue. Again, for those who have not used Jupyter Notebook before, but have installed it and want to continue working along with me, these are the code cells. So to run this code cell, you can click over here on the code cell on the left, such that it highlights with the blue bar, and you can click the run button over here on the top, or you can press Shift and Enter, and that particular code cell will run. So you can follow along. So as we're talking, I'm going to be hitting, I'm going to be running each of these cells so that we don't lose time. So let me run this. So this very first step is to load all the packages and tools that are required for our processing. And so once that is run, you can see that there is a number associated with it, which means that particular cell has already been finished running. Okay, so that's loading all our libraries. And then let's filter the data to contain only the tissue of interest, right? So this is the first one we're going to do is the GTEx data that we just downloaded. And we are going to read all the files that we downloaded. The first one is a metadata sample, the sample metadata, which gives us information about what the sample type is, what kind of a tissue it is from, and whether it is disease tissue or not, and things like that. The second one is the subject metadata, which gives us information about the age of the person, whether it's male or female, and a few other metadata fields. And then the last one is the actual tissue expression data, gene expression data frame itself. What you can see down here is an example or just a snapshot of the top few lines of each of these data sets. The first one over here is the metadata for the sample. Here you can see, for example, this column says which organ it is from, and then there is a more grittier information about that. For example, if it's blood, is it from whole blood? If it's from brain, is it from the cortex, or is it from somewhere else? Things like that. And then there are several other metadata fields. The second one is the subject metadata. Again, as I said, the sex of the individual, the age of the individual is given. And then the last one over here is the rows, the ENSG are the names of the genes. These are the ensemble IDs of the genes that are present. And the second column over here is the gene symbol that's associated with the ensemble ID. The ensemble ID is what is, it's easy to process just because all the data frames have that in it. And so we're just going to use that. But what we are going to do in the next step is take this second column and the first column and associate them to each other. And then we'll just save it so that we can use it later on. So that's what the next block of code does. Just to show you what it processed, we have a total of about 56,200 ensemble IDs, or the row numbers, and we have 17,382 samples in this GTEx data set. The next block, what we're going to do is add the sample metadata and the subject metadata all together in one data frame so that we have everything in one big data frame. So let's run that. And here now you have the subject ID that was in the sample data frame, as well as on the right over here, you can see the sex, the age, and I just converted the sex that it said it was 1 or 2. I converted that to male or female, depending on which number it was. So that's what we've done in the above code block. So we have that now. So I was mentioning that we're going to look at the heart. So to find out what kinds of tissues the data is from, we can look at this particular field that is the SMTS, or it's the tissue site metadata field. So let's see what are the kinds of data that's present. You've got several organs in the tissue in the GTEx data. You've got kidney, liver, blood vessel, uterus, and so on and so forth. Heart, of course, and the brain. So as I said, today we'll look at the heart, and even within that, we're going to look at the atria specifically, because this data set does have atrial samples as well as ventricular samples. So this code, let's run this, and now you can see that we are, sorry, that's the next one, but this one you can see that you've got the heart, atrial appendage, and the left ventricle. And out of the 861 samples that were totally present, they are divided between the two almost equally. And so now let's actually just subset just the heart atrial appendage samples by running this code over here, and we will combine it with the gene expression matrix. So now you have, again, a total of about 56,200 ensemble IDs. Sorry for the word over here, and with the 429 samples. So what you can see over here is the sample name in the first two columns, whether it is male or female in the third column. And then these numbers that you see, some of them are zeros, some of them are huge numbers over here, are for each gene, for each sample, what is the number of counts that was present in the RNA-seq data. So that is given in this expression data matrix. Now, obviously, there are a lot of zeros here or very low numbers. We want to get rid of those so that we can avoid noise in the data and have more specific interactions that we can look for. So to do that, we will filter out those genes that are very low expressed. And we are going to use 3 criteria to do this filtering. We want to keep only genes that have a count of 10 or more in at least 60% of the samples. And when I say 60% samples, I'm talking about 60% of one group of samples. So in our case, we have the male and the female group. So if we have 200 male samples, we will make sure that there is at least 120 samples that have a count of 10 or more for that particular gene. And if it doesn't have that, then we will remove those. So this second the block after that does that. This step does take some time because it's a lot of genes that we are looking through, and we have to compute across them. So let's give it a minute or two. Is everybody following through so far? I think this is probably one of the longest steps for filtering that takes. Okay. While that is going, I'm just going to, not go, I can't go to the next step, but just to say that we will be using the DE-seq package here to do the differential expression. Again, I'm not going to go in detail about that. We will only be using that information, but if you're interested in the tutorial itself, there is a link over here that will take you to the tutorial page, and you can follow through along there. Just a couple more minutes. And this particular step is customizable, right? So depending on the filtering step, I mean, is customizable. So depending on, let's say, how deep you've sequenced it, or how highly expressed your genes that you are looking for, or how how many samples you have in total, or how many groups you have in total, these numbers can be changed. For example, over here, we are saying 10 is the number. So, but you can change that to 5 or 100, depending if all your samples are all your gene counts are very high. You can change this number from 60 to lower. If you're doing it lower, then you've got more noise because less number of samples have to have it. And so it's not as strict. Or if you want to make it more strict and say all my samples have to have it, then this number would become 1. And also, let's say you have several groups in your data set, not just 2 data sets, then you can say, I want one of my groups to have this gene, or I want all of my groups to have this gene. So you would mention that number over here. So that's how you can customize your filtering steps. Okay, so this is done. And what you see over here is now this number of the ensemble IDs have now come down to 24,891 instead of the 56,000. So that has taken out all the genes that had like zeros and ones and things like that. So now let's move on. We will use this counts data and the metadata and form what is called as the DE-seq object. What it does is, essentially just combine all the metadata and the counts and have them all related together. It's called the DE-seq object. Right. Sorry. So that's what it is called. And we will use this for the rest of the steps. Just wanted to point out here that out of the 69 samples, we have about 136 female samples and 293 male samples. Before we even process anything over here, we can generate a PCA plot with the data that's currently present, and we can see what the data naturally, separates out as. The PCA plot will tell us whether there are genes that are segregating the male and female samples. Here is a PCA plot with the male samples represented in the teal color and the female samples represented in the brick orange color. And what you can see over here is there's a slight separation with the female, especially across the PC1 and a little bit on the PC2. And you can see that the female samples are over here and majority of the male samples over here with some overlap, of course, in between. But what are the genes that are causing this difference is what we will look at with the DE-seq portion of it. This step you can run if you know how many cores you have in your computer that you're using to run. If you don't, it's okay. You can still leave the step out. I have, I think, 8 cores, but I'm going to use 4 of them to run this so that we can parallel process, and it's a little bit faster to process. The next step over here is the actual differential expression step that is done using the DE-seq tool. Again, this does take a few minutes depending on how big your data set is. So let's give it a minute or two. Everybody with me so far?
Yeah, we realized based on the replies from the emails, number of people we asked, your basic, your level of experience. And so we have a distribution of skill sets. Some are more experienced and some are new. So yeah, don't be shy about, speaking up, asking a question. Be happy to answer that. There's a question in the chat.
Right. So here, the design model. So that is for the DE-seq, for those who have run that before. So we are using the condition. In this particular case, we are looking for differences between male and female. So I have introduced that condition over here. You can also run this without giving it a condition, a design. So which means you're just taking all the samples together and seeing how it naturally separates. But this one, we are specifically looking for things that are different. I have labeled it as condition, but the condition over here is whether it was male or female. Okay, so let's see. Yes, so that step is done. So the differential expression is done. And so let's look at what that gave us. So it is looking, as I mentioned, it is looking between male and female samples. And there will be several, so for each of the gene that's present, you would get a p-value and a false discovery rate value or a p-adjust value. And so the whole list will be the entire 24,000 genes. But what we are going to concentrate on is to, again, look for only the genes that have a statistical significance. So we want to make sure that we are looking at a cutoff of 0.01 for the alpha, and an FDR value or a p-adjusted value of 0.05. So let's run this step that does that filtering for us. And we will only look at the genes that have passed both those filters, as well as have a log fold change of 2. So that shows us that we have now a list of 66 rows, or in this case, 66 ensemble IDs that were differentially expressed. And these are the parameters that it has. For example, the mean base mean value for each of those samples, each of those genes are listed over here. So you've got a wide range. I mean, some of them are as low as 5, expression value, and some of them are as high as 5,000 counts, right? And then here is your log fold change. A log value of 1 means there's a 2-fold change, and so on and so forth. And then here is your p-adjusted value. Sorry, over here on the right is your p-value and your p-adjusted value. Again, we have already set it to be 0.05, so nothing higher than that would anyway be represented over here. So this is the filtered list. And we have saved each of these throughout. If you go through the code, there are several places where I have actually saved these files. So it will be all in your output folder. And then the next step is an optional step. For those who have this library already installed, we can run it. But if you have not, this is still an optional step. I'm just going to show you how it looks. We are going to generate a heat map for just these 66 genes. And the next code does that. And we're allowing it to cluster by itself. So what you can see over here is all the green samples, in this case, which is the male samples, they all cluster together based on these 66 genes, and all the female samples cluster together. And you can see that each gene has a different expression pattern, whether it's a male sample or a female sample. And again, for those of you who have not seen a heat map before or who are not familiar with it, in this particular case, there is a z-score value over here that shows a dark blue means that there is very low expression, and a dark red value shows that it is very highly expressed in that sample. So, for example, this is the obvious difference between the two sets of samples. You can see a few fairly red, not super red, but fairly red, or maybe closer to white over here, sets of genes that are expressed in the male samples, but they are just blue or dark blue in the female samples, which means they don't exist in the female samples. And there is this one stark example of gene that is expressed only in the female sample and not present in the male sample, right? So this is one way of seeing what the genes look like and how they separate out the samples. Another mode of visualization for these differentially expressed genes would be using, like, box and whisker. The red and blue part, it's not exactly a fold change, but it is the expression values. It's a normalized expression value. So the question asked is the matrix value over here. It's a z-score of how how high the expression is or how low the expression is. It's not the fold change. Okay, so as I said, we can look at the box and whisker plots, and I don't want to look at all the 66 plots if they are not statistically significant. So they are all, because we did filter them out. But if you had further filterings to do, then you can set those up as well. So here we do have a way to represent the genes in, where am I? Here. Yeah, it does take a minute to do the box plot. And here you can set values again. I have kept the same as what we had for the previous step. So I'm going to have all the 66 plots here again. But if you were to change this value, you can then modify it and see for even more stringent gene list if you're interested in that, or relax it for that matter, right? So let's see what that gave. So we have all the genes represented here. And in this case, you have the red, I believe, is the female samples. and the teal is the male samples, which is what we saw in the PCA plot above. And these two genes are higher in the female samples. Again, we did see one gene that was higher in the female samples in the heat map, or at least starkly different, and that's probably this exist. And what you note, I don't know whether you noticed it over here, is from the ensemble IDs, we now moved to the okay, I'm going to come to you, Andrew, just in a second. So what you can notice over here is that now they are not ensemble IDs, they are now gene names. Gene symbols is just because it's easier for us to understand what gene we are looking at rather than ensemble IDs, which are not very intuitive for for us. But to answer Andrew's question, which asks why are we not using the male and female in the model, we are using the male. I'm hoping that you're asking about the DE-seq design model, right? So there I did use the word condition, but the condition is whether it was male or female. So let me go back to that step just to show. Let's see, where's the DE-seq object here? So this is the DE-seq object, and my design model contains condition, and I have denoted what condition means. In our case, the condition means whether it was male or female. Okay. Okay, so here is a list of all these genes, right? So some of them are higher in female, and then most of these seem to be higher in male samples. That's all good. I know these plots are all super small and hard to read, but if you wanted to print out each of these, you can use this second block over here to print out each one in a separate PNG file, and that will be saved to your results folder. I can run it just for the sake of showing you, but we are not going to see any output over here on this page, but your folder, if you're running this along with me, you should now see your results folder populate with several PNG files. And it does take a minute again because we do have 66 plots to put up over here. Okay, so that's done. So any questions other than what has already been asked in the chat? We can take a break here because the next section is going to be pretty interesting as well.
We're pretty good on time, so yeah, I'm sure there's some questions. Feel free to speak up. You can unmute yourself if you want to and ask.
Yeah, so Ahmed has asked, do the ENSG and the symbols have a one-to-one relationship? Yes and no. So the ENSG does come in two forms. So one is the actual, let's see whether I can go up and show you what the ENSG looks like. Here, no, this one, right? Okay, so you've got this ENSG that has a huge number, right? ENSG 0000, and then a huge number, and then you've got a decimal point and a number after the decimal point. The ENSG ID that is before the decimal point has a one-to-one relationship with the gene symbol, yes. But once you have the decimal point, then you have several ENS IDs that are associated with the same gene symbol as well. So yes, when you're doing filtering of the data or coming up with these things, you do have to be aware of that and address that depending on what your condition is. I believe the decimal points are just for different isoforms of the samples or different fragments that have been picked up by some method or the other. Can you summarize what we have done? Yes, I can. So let's go back to where we started. So this is our overview. So far, we have just done the first portion over here. We went to the GTEx website, downloaded the data from there. We had to download 3 different things. One was the actual gene expression matrix. Second was the metadata. And within the metadata, there were 4 files, I know, but 2 of them actually have the metadata. And 2 of those files had the explanation of what each field in the metadata file was. So we downloaded all those 4 metadata files. Then we read all the 3 files, actually, the gene expression matrix, the sample metadata, and the subject metadata. And then we filtered out all the tissues that we were not interested in and only kept the heart tissue and specifically the atrial samples. We were not looking for the ventricular samples. Again, if you want to try this out with a different tissue, you're more than welcome to try it out. But today we're just doing the heart. And then once we have the filtered tissue, well, during the filtering, we also did a removal of genes that were very low expressed and kept only genes that were expressed in, at a count of 10 or more in 60% of either the female samples or the male samples. And then we normalized the data, ran DESeq2 to do a differential expression of the genes that we are specifically looking for. So in this case, male or female. And then we looked at the heat map as well as box and whisker plots to look at genes that were differentially expressed. And what we noticed was that majority of the genes were highly expressed in male samples, and just a couple of genes were highly expressed in female samples. One thing I wanted to point out was that, again, I know that these ensemble IDs are not very helpful, but if you see over here, all the genes that are highly expressed in male, most of them have this Y tag that just means that it's on the Y chromosome. And not all of it, but majority, it means that it's on the Y chromosome. And this exist or the SIX, which is just the opposite strand of the exist is they are both present in the X chromosome, and as expected, they will be either high or low in the female, depending on the sex of the individual. Is that a good summary? Matt, should I continue or should we take a
Yeah, you could, maybe you could ask for kind of some people are following along and some people are observing, I think. So maybe you could ask for a thumbs up if where people are at. Does it continue, or how about a thumbs up if you're been able to follow along okay? We're seeing mostly thumbs up, so that's good. Another
Is there
question?
a thumbs down?
Another question?
Okay, some of the differentially expressed genes have NS. Yeah, I tried to figure out these all should be different. These all should be significantly different because I did. There is here, right? So this file that you should have a file that says GTEx significant genes, which is those 66 genes. So if you look at the actual file, actually all of them do have the p-values that are significant. I don't know why they're showing up as NS over here. I tried fixing it. I couldn't. So yes, for example, these two genes over here does say NS, and I don't have an answer for that, sorry. But the p-values in the Excel file does show that they are significant.
I think you can probably go ahead and continue, it seems like.
Okay. Yeah. Okay, so that was the first part. The second part is pretty short in our case. We will be looking at, and as I'm talking, I'm going to actually run this cell because, again, it does take a few minutes for it to read this cell. So let's first run that. And what I wanted to tell you was that we have chosen this particular library, MAR-TAR-RNA-seq, to study these interactions and to identify the different targets. And as I mentioned in the beginning of the workshop, this is a database where it looks at all the interactions between the miRNA and the mRNA that are annotated. It's all there. So I think we have like close to 2 million different combinations of these, or maybe more. But these are not experimentally verified. There are other databases that have experimentally verified interactions as well. So if you want to use those, you're welcome to try those as well. But here we are just using the predictions based on the interactions. And what it gives you is data frame that looks like this. So this is what, if you actually use the package, this is what you would eventually get. It lists the microRNAs. Of course, this is just the top few, and this lists the microRNAs and the ensemble IDs. And again, as you somebody asked, the ensemble IDs do not have the decimal point over here because there will be too many of the repetitions over here. So we just want just the ensemble ID without the decimal point. And then it has all these different fields that tell you, where it binds or what specific spots it binds to, and what is the folding energy or how well it interacts with that gene. And the greater the negativity of this number, the stronger the interaction is. Okay, so what you can, and I have also listed out over here a few different information that's from this data set. What's the threshold is, again, the threshold is, I think, how many different, what is it again? I forgot. I know I looked it up. I'm sorry, I don't have an answer for that right away, but that is a cutoff I used to get this data frame. I think that threshold ranges anywhere from like 20 all the way to 140, 150, I believe. And I only looked at, I think it's the confidence that they have for that interaction. I think that's what it is, but I'm sorry, I don't have the exact definition of what threshold is. But what we are looking over here is only those that have that high confidence value, which is above 140, which is what I said. Again, that can be changed. Okay, so with that, if you look at the number of microRNAs that are present in this data frame, we have a total of 2,656 different microRNAs, and there are 19,247 unique combinations that are present. Unique mRNAs that are present. And then if you look at the number of interactions that are present, you've got, I think, what is that? That's about 24 million different interactions, right? So we don't need all of these, and not all of these are strong. So, and the range of the folding energy goes all the way from negative 1.78 to negative 45.9. So we, in this code, I have said only look for folding energies that are lower than negative 20. And so when we do that, we finally come up with 10,000 interactions for the combinations. And then of those, we have 1,982 microRNAs that are unique. Okay, so that was the second part. So we're just using it based on this data frame that we got from Miranda. And we can move on to the third section. So the third section is looking for small RNA-seq data from the xRNA atlas. Again, there is a hot link to the atlas, and I also have screenshots on how to access the data from that. But let's just go to the website right now and see how to do this. So let's click on that, and it takes us to an interactive page where you can choose different filters depending on what your analysis is looking for. In this case, we are going to be looking for healthy samples, healthy controls. So now that reduced the total number. If I didn't have that, I think we have like more than, I think 10,000 samples or something like that. I might be wrong on that. But if I select healthy samples, I have 3,534 selected. And then let's say I want to just look at the plasma samples. Now we reduce that to 2,200. And you can look at different types of RNA source, so extracellular vesicle or an exosome or H-tail complex. But today we'll just look at the total cell-free biofluid RNA. So now that's reduced the number to 1,712. And then again, there are several isolation methods that were used, and I'm going to pick, let's say, the serum and plasma QIAGEN kit. So that now leaves us with 456 samples. You can then press on this magnifying glass or the search tool over here. That takes us to the next page. And it pulls up all the samples that were there in this 400 and odd samples. And it gives you lots of details about what those samples are. the first few samples over here, you can see this is the biosample name, and it tells you whether it was a healthy sample or not, plasma, what method of isolation, and things like that. And it also tells you what assay method was used. So obviously this is qPCR, and I'm not interested in qPCR at this point, only at smaller NA-seq. So I'm going to keep going down to see if I have any smaller NA-seq, and yes, I do have smaller NA-seq over here. And it looks like the file names are JFREE, and then that's a long list of files that are present. There are other things also that's not JFREE that seems to be having a slightly different naming convention. And so what we're going to do is, and then we also have over here on the right is this accession ID and this FASTQ identifier. I think these are the two main fields that we are interested in. So to download all of this, you can go to download all of this, we go all the way to the left. Select all the samples. Again, I'm sorry, I know I'm walking through this. You do not need to do this currently because all this data is already downloaded and you have it in your Google Drive. But of course, if you want to follow through, you can. I'm going to have to deselect all of these samples, that are not of interest to me. Let's say I'm just deselecting these samples just to show how to do it. And then you can click on this download metadata files and download the biosample metadata files over here. Okay. So now that would download. I'm going back to the Jupyter Notebook. You can download it, as I said, but if not, the data is already there. So let's go back to the Jupyter Notebook. And that's what I've shown here in screenshots on how to download. And what we are interested in mainly is this accession ID, and I'll tell you why we need that. So the file that you download will look something like this. So the first portion is going to be the same, and then depending on when you downloaded it, this value is going to change. And it contains several fields, of which we are interested in these 4 fields. Just as a reminder, when you actually download it, right now you don't have to do anything with the file that I had provided. But if you downloaded it, I just want to remind you that it's a CSV or a TSV file. And there's a header section that's about 76 rows. So you have to delete all of those first 76 rows and then resave it into the same current folder that you are working with under the subdirectory ERCC for the rest of the code to work. So and that's just because all that is just information about how this consortium is and how the data is present and things like that. So that is not going to go into our analysis. So once you delete those 76 rows and resave it, we can then read those files. So I'm going to read the file, and what it looks like is this. So you've got the first column with your the FASTQ identifier, which is the name of the FASTQ file. And then you've got the name of the sample, which is the biosample name, and then the condition, the biofluid, and all the other information that was present. And here is your accession ID. I don't need all of this, and I just wanted to point out one other column over here that says meets ERCC QC standards. What we're going to do in the next step is anything that had a fail in this column, I'm going to remove that, and I'm going to reduce this metadata to just a few columns instead of having all these columns. So that's what we'll do in the next step. And so now it's a cleaned up metadata file where we have, just to point out over here.
You have a question in the chat?
Can you just skip those lines when you, I guess you can, but this particular code that I have does not do it. So yes, if you know how to skip those lines when you read the file, yes, you may just skip them. You don't need to delete them. But this code that I've written takes into account that you've deleted it. Okay, so if you see the original file, it had a total of 398 samples and 17 columns of metadata. Now, with taking out those failed samples, you now have 383 samples with 5 metadata columns. And I know we've already filtered out for things that we want, but in case your analysis set has a lot more data, then we can run this thing to see how many different biofluids were represented in this data set and how many were, what kind of a condition it was the samples were. So here, all our samples are plasma and all our samples are healthy controls. Okay, so we're just going to save this exact data frame into several different smaller files because I'm going to use that for several for different purposes downstream. So we're just writing different versions of the same thing. And this is the portion that takes a little bit of back and forth between the terminal and your data and the analysis set that you're looking for. And I'm not going to walk through every single step. I can give you an outline. And this is because, again, of I'm not sure how many can actually run it. And if all of us run it at the same time, maybe we will hang up the portal at the same time. So I didn't want to go through that during our presentation. But the expression matrix that we need for our study has already been downloaded, and it's provided to you in that link under the subdirectory ERCC. But if you were to, let's say, on a different day, you want to download a different analysis data set, and you were looking for some other, let's say, disease versus a control condition, and all those kinds of data are present in the ERCC. So if you wanted to do that, you will run this set of code. So these just there are four codes that you have to run. The first one is getting the analysis.json file that contains all the biosample names that's present in that analysis. Then the second one over here is to get the individual files for each biosample that was listed in the analysis document. So if you had 383 samples, then you would have 383 JSON files. Just so we see it, I just want to show over here. I think we already have that over here. So for example, over here, all those are already downloaded over here for you. So you do not need to download it currently. But if you were to do this on your own, you would run this script to be able to download all those. So again, it does take time because depending on how many samples are there in your analysis, that many downloads will be there. And then you will subset it to just look for, to get the male and female. So each of those biosample JSON files will have information about the biosample. And so in this case, we are looking for male or female. And so you can pick out only those things that have male and female information in it. And then ERCC, or the xRNA Atlas, has, for each analysis, they have an entire a CSV file that has all the samples that were analyzed in that data set put together as a miRNA expression matrix. And so you will download that, but then in the following steps down that I'll show you now, you will subset them to only care about the few that you are interested in your analysis. I know this is a little bit, unless you do it, you're not going to probably know exactly what it does, but I just wanted to let you know all these files are present. So these are all scripts that I have written to be able to do these downloads. They're all present in your ERCC folder. So here are all those PI documents, SH, and your scripts. You can look those up and see and modify them as you need it. I have written it such that they're looking for healthy controls and plasma samples and get the male or female information from it. But those are the places that you can modify. And I also do have, I'm going to try and open this so that you can see that as well. Sorry. So this is, if you were to modify those scripts, of course, you can do it directly in the script, but this is what the scripts look like. And you can change information. Let's say over here, let's say you want to look for healthy control versus, I don't know, Alzheimer's disease, right? So then you can type in that over here, and then now your subset will contain healthy control as well as Alzheimer's disease conditions that are present. And then again, I have said plasma, but let's say you wanted to look at CSF in that case, if you're looking for Alzheimer's, then this would be cerebrospinal fluid or CSF, depending on what was the actual terminology that was used in the biosample thing. So here you can edit those, and then if you run this cell, you will get a new script file that you can then use to download your samples of interest. Okay, so I know that was a lot of information that we are not running through. So you can reach out to me later on if you needed help with that. We are not going to go through that particular code in detail. at the end of all this, the files that I'm actually going to be mainly interested in are these two files, actually. So the healthy plasma text, which is a subset of the file of the samples that we're looking for, and the microRNA counts, read counts file, which is your gene expression matrix for all the samples that were present in the analysis. Any questions for this download portion? Other than how to run the script, which we can go separately if you need it.
And feel free to
Okay.
unmute yourself if you have a question.
Yeah. Yes, please. Okay. So if you don't have any further questions, let's actually now, oh, there. Should we apply batch correction? For the DEC portion, are you talking? Han Kim? Yes. I guess it depends on how the data would look like. So originally, when I ran this, I ran it on all the heart tissues together. So as I said, there were atrial tissues as well as ventricular samples. And so at that time, I had a trouble with how the data looked like on my PCA. So I landed up doing a batch correction for the organ at that point. But yeah, so depending on what it looks like, so you would download the data, do a PCA analysis, I guess, and see how it looks like. And if you think that there is a batch effect, then you can do that by doing, I'm going back to the DESeq just for people who have not seen it before. So you would go back here where you create the DESeq object. And here you would add plus, let's say, I'm not going to have it over here because I don't, I'm not doing it. But if you do SMSTD, which was the specific, site, then you would do that over here. But we're not changing that for our current analysis. Okay. Where are we? Okay, so we downloaded everything. We have all our files, and we have filtered out things that we don't need. Okay. Okay, so now let's actually do the data processing, and it's going to be very similar to what we did for the GTEx. The same steps will follow. So let's first read the metadata file. So we're going to first read the metadata file in this case and make sure that it matches the subset that we did from the the ERCC API thing. So what this just says is that I have all the samples represented and they all match each other in their order, so that we don't mess up the order when we do analysis. And we have now, the name of the sample, the sex of the sample, and the FASTQ identifier, as well as the type of sample. Okay. Then we are going to read the actual counts matrix. And again, this will be now subset to contain only these samples that were there. So let's run that. And I believe I don't have any output for that. You can. So all of these code has output commands. I have just silenced it because I didn't want it to populate so much. But you can always just take out a given hashtag, and that if you run it again, then you will have what it looks like, right? So I'm going to keep this over here. And then this is the step again that we are doing filtering for the microRNA now, which again have a reasonable expression. So we are again keeping a number of 10 to be the minimum number of counts in at least 60% of the samples in at least one group. That's what we are calling it. So it's the same conditions as before. And as I said earlier, you can change it as per required. So let's run this. And this particular one will not take too long because it's a very small data set in the sense you don't have 56,000 genes to look at. We are only looking at about 2,000 genes. So that went pretty fast. And now we will create a DESeq object. And so like others were asking earlier, in the previous one for the GTEx portion, this design was used to include or look for genes that were specifically differentially expressed between male and female. You could do that here as well, but what I'm choosing to do is I'm not looking for differentially expressed genes for this data set. And the reason was actually because I've already tried it out and I didn't see any differences, and that's why I didn't do it. But if you just wanted to see what the data looked like without doing any modifications to the data, then you would do a design tilde1 that just says, don't look at anything specifically, just look at what is there and tell me what is there, right? So that's what we're doing for this particular small RNA-seq data. So let's run that. And what you can see here is it tells you it has 383 samples with 48 different microRNA. Interesting. This microRNA count was from, where was it? I think I didn't mention that over here. This has removed the microRNA this has only kept the microRNA that is present in the DE-seq that we did for the miRNA, mRNA target that we did. So at the end of that, whatever microRNA is in this data set that was present in that target list are the only ones that are kept here. So we are only looking at those microRNAs. Okay, so that's the filtering that we've done over here. And then we're just going to look at what the PCA plot looks like for this. And in contrast to what we saw for the GTEx, there are no male and female differences that are prominent in this small RNA-seq data. Okay, so we will now normalize the data using DE-seq2. And we are going to look at the expression of we're going to do box and whisker plots, just like how we did for the GTEx things, for just the microRNAs that were present in that data set. And now we want to make sure, since we are not doing differential expression over here, we don't already have the significance values. So we're going to calculate the significance values again over here. The setup is the same, and we want to see which of the microRNAs are significantly different between male and female that are also associated with a given differentially expressed gene. Okay, so that we'll run that. And for this data set, it looks like there are 3 microRNAs that, from the looks of it, these two, the 652 and the 122.5p, are higher in female samples, in male samples, and the 4433b5p is higher in the female samples compared to the male samples. Again, you can save these plots if you want separately. And of course, this thing can change depending on how many microRNAs were different in your data set. And then we are going back now to the differentially expressed genes and seeing which of these genes are actually associated with which of these microRNAs. So these 3 microRNAs specifically, which are the target genes for these microRNAs? So we're going back to the Miranda data frame that we had with the interactions and going to pull those out. So looks like the genes that are present, that are differentially expressed in the male and female samples, are these 6 genes. And so I'm going to leave you with this, because now at this point, what we will do, what you can do is, now you have a list of genes that are differentially expressed and have strong differences in the microRNA expression levels as well, that we can then go and look at these genes and see how they are associated, or how they are functionally different in male and female maybe, and make our own analysis based on that. So this is the overview for the use case today. I just wanted to say that we have taken male and female as a sample example, as an example of how you can do comparisons. But both in the GTEx data, in the GTEx data, the data is all from normal tissues. But in the xRNA Atlas, we have data from several diseases and several biofluids. So you could do similar analysis with different data sets or with different conditions of your interest, and you will find more interesting genes or more interesting patterns. You could also do something very similar with, if you know how to do small RNA-seq, for example, I'm sorry, not small RNA. If you know how to do single RNA-seq, you could look for genes that are differentially expressed between male and female, and then go to the HubMap uh, data coordination center, get the data for the single cell RNA-seq, let's say from the heart, and see which of those individual cells contribute to those differences that were present in the bulk RNA-seq. So those are examples of how you could analyze, how you could use the different data coordination centers to do analysis across data centers. That's all I had to share. I can take questions, or if you want me to go over any part of this again, I can go through that.
Yeah, maybe you go to the part 3 and kind of summarize again what you went through. But yeah, please do speak up. Feel free to unmute and ask questions. And also just put a link in the chat. Your feedback in the workshop would be greatly appreciated. It'll only take a few minutes. This will help us improve CFDE resources in future workshops. So yeah, we'd appreciate just a couple minutes of your time. But yeah, it's feel free. It's open forum now to ask questions. And if you're got stuck on any step along the way, you can go through that. So maybe you could just summarize what you went through and then open the floor for questions.
Yeah, so the very last portion, which is the part 3, was the xRNA atlas data. So this is the extracellular small RNA data. One thing that I didn't mention is the xRNA atlas also. So what I showed was the microRNA expression matrix, but you do have, the gen code or the protein coding section of the thing that is also being mapped to. So you do have that data as well. And you do have several other small RNA forms that is present over there, like tRNA, piRNA. So any other interest can also be done. But what we did today was to download the microRNA expression matrix and the associated metadata, and then filter the data to contain only microRNA that were reasonably expressed, in this case, at 10 counts in at least 60% of the samples. And we were looking specifically for healthy plasma control samples in our case. And then we normalized the expression data. We did not do a DE-seq2 itself, in the sense we didn't do a differential expression for the small RNA-seq. We only normalized the data. And then we visualize the expression patterns using a Boxen whisker plot. And that was specific to the genes that were associated, that were differentially expressed in the first phase. So yeah, please do. I'm here until you need me to be, and I can walk through any steps if you needed help with that.
I just want to mention anyone who's interested in using data from the xRNA Atlas, there's just over 10,000 samples there now. We are in the process of adding a few thousand more samples that should be there by the end of this year if you're interested in that data set.
Correct me if I'm wrong, Matt. I think we'll also have long RNA-seq data as well from extracellular RNA.
That's right. There's some long RNA-seq data will be deposited. Yep.
Yeah. So then you can compare long RNA-seq from tissues and from the plasma or serum samples as well, and then compare those.
We would be curious if anyone's willing to share if you have actually used GTEx or ERCC data previously, or if you're looking to combine with some of your data. We'd just be curious if that's something that you're considering or have done or are considering. Would anybody be willing to comment on that?
Can anyone have access to all the databases? Any review process is required? I think there's a reasonable amount of public access data in all of these data coordination centers.
Yep.
But there are protected access as well. So depending on what data set you need to access, I think there's quite a bit of information, quite a bit of data sets that you can access without, because they're all published and they're already present over there. So it should be possible. But
Yeah.
some of them might require.
Maybe you
Sorry.
could go because it was at the start, but maybe go back to the GTEx where you show there's open access and
Oh
um,
yes.
it's the same thing with
Let's.
Empress. There's some that we have links out to the GRDB gap but most of the data there you can just access from the Atlas. GTEx is similar. And then maybe you can also, yeah, so you can talk about that. Then maybe you can also go to the CFD workbench where you can do the metadata search across all the 10 or 12 different GCCs, but you could show both of those as a reminder.
Yeah. So I guess I was trying to answer. Okay, sorry. Yeah, let me go to the CFDE workbench, but somebody has asked for the Jupyter link, a Jupyter Notebook link. Yes, actually, the CFDE, the GTEx specifically does have the aging brain. Yes, I can show you where they are. Just give me 1 minute. Somebody asked for the link for the, David, would you be able to give the link to the Jupyter Notebook that's present in the chat, maybe? I don't know. Okay, so you've got the workbench over here, and what there's already. So sorry, let me go back. So the GTEx portal does have an open access data over here, right? So that's where we went, and the because the metadata has the age as well for the individuals, you can look at the brain in the GTEx data for looking for differences. But what I did notice was that in the CFDE data portal, if you go to the data portal itself, you can search for GTEx. And here they have, for example, aging signatures. There's already analysis that's done for several tissues with respect to the age. So I did notice, I think if I searched for a brain, I might get it. But I think I did notice that there was, let's see. Let's search GTEx brain. Yeah, so here. So you do have analysis. It's already been done. So you might be able to look at uh, the analysis from here. Um, yeah, there's plenty of different metadata and process data sets. So this is another place that you can go to. Here is the process data sets that you can click on, and each data coordination center. For example, this links is a good data coordination center that actually contains the effect of drugs on on the expression of different proteins and RNA that is present. And so that's if you're looking at the effect of a drug-induced problem or the treatment effects, that's a good data set to go to. And then you've got Kids First Data Center, data coordination center. I think that works with cancer in children, if I'm right. And so there are several data sets that you can look at them. And so you've got different types of digital data that's present as well. And they've got all the tools and workflows over here. They also have webinars that are present, so you can look through them for more detailed information. There's quite a few videos present to go through this. Anything else, Matt?
Yeah, I'll just give people a second. Any questions on the process or any technical issues you encountered or anything about the data sets?
Okay, a question is, when you filtered low count genes, you did that in a condition-specific matter? Is that what you normally recommend? No. I do agree that you should, first filter out all the genes that are low expressed and then do condition-specific things. So let's go back and look at what I actually did here. That's my metadata. That's my heart. Uh, that's my heart. No, so here I did do the selection first. So I'm not looking at in my case, male and female conditions is the different conditions that I'm looking at. So this step, which is the filtering step to take out low expressed genes, is done before my DE-seq object creation. So whatever low expressed genes were there, they were taken off before, and now we are only looking at consistently expressed genes to then represent the different conditions. So you should do the filtering before you look at your conditions.
I'm just curious, can anyone give a thumbs up if you kind of downloaded the Jupyter Notebook and kind of went through the process yourself with the data? Anyone do that, whether before or after or during the workshop? It's like both are following along, which is fine. Oh, there's one. Okay. Okay, there's a couple. Okay, good. Has anyone used CFD data besides, or that's not GTEx or ERCC? Has anyone used other data sets from CFDE? Okay, any other questions about anything about the process or the data sets? Okay, I guess if not, Mina, the recording will be available. Feel
Yes.
free to access that or share that. We'll send you an email when that's available. Again, thank you for attending. We appreciate all your comments and feedback, and do appreciate if you could just take a couple minutes to fill out that survey. That would give us some great feedback, I'm sure. So thanks to everyone. Thank you. Thank you, Mina.
Thank you so much for the opportunity. And yes, please don't hesitate to contact us if you need anything about this.